So I'd like to welcome you all on behalf of the Johns Hopkins Center for AIDS Research and the Department of Molecular and Comparative Pathobiology. I'm probably one of the few people that can say it because I named that department. And it's really my great pleasure to introduce Beatrice Hahn. I've been in the field a long enough time to remember when she entered the field. She's always been, from the very start, a bold leader in this field. She's done things. Her papers are always something new, something that other people haven't done. She's explored areas in HIV/AIDS that have really changed the field. Dr. Hahn received her MD from the University of Munich and did her internship there. And she then came to the NIH, and she, at the NCI, she joined the lab of Bob Gallo as a postdoc, and that was in 1982. And so she was in that lab at the beginning of the HIV epidemic. And her very first papers from Dr. Gallo's lab, her very first paper was in Nature. It was on HTLV1, but it was in Nature. And then that was sort of set the tone for her career, I think, when I look through. And her next paper was on the molecular cloning and characterization of what was called then HTLV3, which we now know as HIV-1. As well as the other thing that is very notable to me and many of the people here that are interested in HIV, she had the first report of HIV in the brain of children and adults with HIV encephalitis. And she also followed that study up with a report that HIV not only infected CD4 cells, but T cells. She is really known for her work on showing the first, her lab, along with George Hahn, to show an understanding of the extensive in vivo genetic variability of HIV. And we know how important that is. It's important for everything about the virus, the disease, the pathogenesis. And discovered that recombination between these highly divergent viruses is the major driving force of both HIV and SIV diversity. The work that she's going to talk about today was also, as I say, bold and really exciting. She had to develop the methodology. She had to go where other people did not want to go. She demonstrated the origin of HIV from specific species of chimpanzees in landmark studies and has provided a broad understanding of the emergence of HIV from African non-human primates. As I said, her research has always been novel, and she has always led this field. Dr. Hahn, after she was at the NIH, went to University of Alabama, and she was there for many, many years, from 1985 to 2011. But we've been very lucky to have her now at the University of Pennsylvania so that she can get on a train and come down and be with us. So I just wanted to welcome Beatrice and say we're all looking forward to hearing about your work.
Thank you very much for the kind introduction. It's interesting. I'm looking at a 30-year career, and when I started out, I was really this blue-eyed, bushy-tailed European postdoc who didn't know anything. And fortunately, I met my husband, who taught me many things, and he was also the one who actually discovered HIV in the brain. It wasn't me. We were together on the paper. But to have him now referred to as George Hahn is the ultimate validation that women can make it in this field. So, as Janet just told you, we've known for some time that HIV-1, cause of AIDS, originated in chimpanzees. And a couple years back, we published that Plasmodium falciparum, the cause of malignant malaria, originated in gorillas. And so two of the most widespread and virulent diseases of modern humans originated in apes. And what I'm going to tell you today is there's a third one, as of today, published in Nature Communication. Plasmodium vivax, the second bad Plasmodium parasite, also is of African ape origin. And so it's obvious to ask what we can learn by comparing and contrasting the origins of AIDS and malaria parasites with respect to gauging the risk of future zoonoses. So why bother? Why study the zoonotic origins of anything, including AIDS and malaria? Well, obviously, pathogens must interact with a large number of host proteins in order to replicate, and thus tend to cross more readily between genetically closely related hosts, which of course apes and humans are. But despite substantial exposure, humans appear to have been successfully colonized both by HIV-1 and Plasmodium falciparum only once. And so that tells you that even ape pathogens have to overcome substantial adaptive hurdles to become established in humans. And although the precursors of HIV-1 have been transmitted, as I will show you on multiple occasions, by successfully colonizing, I mean the AIDS pandemic, and that only occurred once. And so we always felt that characterizing these hurdles that these pathogens have to overcome may identify vulnerabilities that could potentially be exploited. So just to get everybody on the same page, what are we talking about? First part of my talk is about primate lentiviruses, of which HIV obviously is one, and then the second part of the talk is devoted to malaria. So we've known for a long time that HIV-1 and the second AIDS virus, HIV-2, belong to a group of viruses that are called primate lentiviruses. And I think Janet was one of the very first to look at lentiviruses in sheep and goats 30 years back. And these viruses are called lentiviruses because their disease takes a long time to develop. And there are other lentiviruses from, as you can see here, other different species, but the primate lentiviruses form one group in this phylogenetic tree. And I'm going to show you a bunch of these trees, so you should know how to read them. Time flows from left to right. And if a branch is long and then there is a cluster after it, that usually means the cluster is significant, meaning that these sequences belong together. And that's certainly true for the HIVs and SIVs. But what you see inside the group is that HIV-1 and HIV-2 don't cluster together, but they cluster in the case of HIV-1 with a chimp lentivirus, and in the case of HIV-2 with an SIV from Pseudomangabes. And that in and of itself tells you that they are derived from different primate hosts and from different cross-species transmission events. So how do you study endangered primates and apes in particular? And really the reason why we know so little about infectious diseases of apes is because they're not accessible. They live in remote forest regions. They're highly endangered. They're protected. You cannot get close. If you do, it might be dangerous. So the only thing you can really do is try to collect non-invasively either urine or fecal samples, and in our case, it's the fecal samples that matter. And then you use those samples for diagnostic purposes. And in the case of SIV, we learned the way we preserve these is in RNA-LATA. RNA-LATA is a high salt solution. You can dialyze the material once it's in the lab, and you can reconstitute the antibodies, and you can actually run Western blots. These are Western blots, where you have viral proteins separated on strips, and then you react that with the fecal extracts, and you can actually identify antibodies just like you would for an AIDS test in people. And so that is a great diagnostic tool we have, but it doesn't stop there. You can extract nucleic acids, either RNA or DNA. In the case of SIV, of course, we are interested in RNA because that's an RNA virus. You can amplify it after reverse transcription, either partial or full length. You can do the types of phylogenetic analysis that I will show you. You can even reconstruct infectious molecular clones by piecing them back together. And if you're interested to confirm the host species, you can get mitochondrial DNA extracted, and you can tell this is a chimp or a bonobo or a gorilla, or even the subspecies of them. And you can enumerate individuals. If you're out there and just collect fecal samples, the chimps or gorillas don't come attached. And so if you want to know how many individuals you were sampling, you need to do microsatellite analysis, and that is possible. So we went out there, and with an army of collaborators, mostly primatologists who had their own interests studying wild apes, we collected over 10,000 fecal samples. And they are from field sites that you see here throughout sub-Saharan Africa. And what I'm showing you here is the sites that are positive for SIV. These are true field sites. They are locations in the forest where you see the fecal sample, but not the chimp or gorilla that deposited it. And you see that the distribution of SIV is actually not across all of the 4 chimpanzee subspecies. Only 2, the central chimp and the eastern chimp, are naturally infected, whereas the Nigerian Cameroonian chimp and the western chimp are not. And there's a second species, the bonobo, that is also not infected. And there's actually a reason why this is, and the simple reason is chimps acquired their infection after the subspecies and the other species had already diverged and were physically separated. So that tells you that SIV in chimps, similar to HIV in humans, is a more recent introduction. We looked at fecal prevalences and saw that they range anywhere from 5% to 15%, and since these are fecal prevalences, the likelihood that we missed something is high. So these infections are endemic in these wild populations, and they're well established. And the fact that Western chimps were negative actually explained why all the chimps in American and European primate centers and even sanctuaries, why they were negative for SIV. Because most of them came from West Africa and the virus never made it there naturally. Now, 10 years ago, Liz Bales from Paul Sharp's lab actually looked at the genome of the chimpanzee virus and realized it was a recombinant. It was a recombinant of portions of viruses that infect prey species. These are monkeys that chimps like to eat. And the chimp virus is a recombinant of 2 of those. The 5' half here comes from an SIV that's called red-cap mangabe, and the 3' region here that includes the envelope, TAD, and REV comes from a lineage of SIVs that infect greater spot-nosed monkeys, moose-dust monkeys, and Mona monkeys. And so some time ago, chimps preyed on these monkeys. They acquired these infections, and a recombinant was generated, which is now the infection of the chimpanzees. So every chimpanzee naturally infected has this recombinant. And this will become important later on. Same with gorillas. Gorillas, there are 2 species, Western gorillas here in green, and Eastern gorillas here in brown. And again, we screened primarily Martine Peters, my collaborators, did all the gorilla sampling and screening, and we found SIV gorilla, and I'll show you where this virus had its origin. And we found it to infect Western but not Eastern gorillas, although our sampling was not as extensive in Eastern gorillas as in Western gorillas. So putting all these viruses and their sequences together, this is the family history of this group of viruses. What I'm showing you here in black are chimpanzee viruses. And what I show you in color are incidences of cross-species transmission. So the first thing you see is there are 2 major lineages, what I labeled here PTT and PTS. These viruses, as a group, infect central chimps in West Central Africa, or Pan-Troglodytes troglodytes. These viruses here infect eastern chimps, or Pan-Troglodytes reinforthii. And they are separate. And the next thing you see is that the cross-species transmission have all occurred out of this group, not this group. And the cross-species transmissions have gone to humans. This is the major group of HIV-1. And as you can see here, there are 4 groups, M, N, O, and P. These are 4 separate cross-species transmission of ape viruses to humans. And then you see the gorilla virus here, which is also cross-species transmission, where virus went from chimps to gorillas. And the position in the tree also tells you the host, or the original source. So we know that group M, which is the cause of the pandemic, and group N, both originated in chimps. We know the gorilla virus came from chimps. And we know that group P, and next week at CROI, our annual AIDS meeting, you will hear from Martine Peters' group, both O and P are from gorillas. So 2 transmissions from chimps to humans, generating M and N, 2 transmissions from gorillas to humans, generating O and P. 1 transmission from chimp to gorillas, generating SIV-GOR. And interesting, and you of course know the epidemiology of this. Only group M has gone pandemic. N is very rare, just about 20 individuals known, all from Cameroon. SIV gore is relatively rare. Group P, only 2 individuals with that infection have ever been identified. And Group M is epidemic, but restricted to West Central Africa, Cameroon, Nigeria, neighboring countries. And interestingly enough, none of these Reinforthia viruses have ever made it into humans. And that surprised us. Now, you also know that there are 40 primate species out there, all in sub-Saharan Africa, that have their own version of SIV. Sykes monkeys, colobus, mandrills, you name it. They all have their separate versions of SIV. I've just told you that gorillas and chimps have transmitted to humans and generated HIV-1. And here are the Sudi-mangabes from West Africa that were transmitted to humans and have generated HIV-2. And then, of course, SIV-MAC in macaques is an inadvertent transmission of a Sudi-mangabe virus in a primate center. But why don't we have HIV 4, 5, 6, 16? And the reason is this: restriction factors. Turns out species have evolved innate immunity to protect their organisms and cells from invaders. And these are examples of interferon-stimulated gene products that are there to fend off whatever bad bug wants to come in. And there are a whole host of these, and the ones on the slide have anti-HIV activity. There are others that have activity against other viruses. And there are still others that probably haven't even been discovered. And they're quite potent. And the ones that I want to focus on today are Apobec and Tethering. And I'll show you why. So I told you that the chimp virus was a recombinant of a red cap-mangabe virus and an ancestor of these guanine viruses. But when you look at the genomic organization, this could have been a simple transmission of the envelope cassette from this lineage into basically the backbone from this lineage. But what you will see is that this is not the case because there's one gene, the VPX gene, that's missing here. Why is that missing? There was no reason. The recombination process didn't necessitate that VPX was missing. So why is VPX missing? So we worked with Mike Emmerman and Lucy Etienne to look at that, and initially pursued a couple of different questions. Well, maybe the ancestral red cap-mangabe virus didn't have a VPX. Well, there's no evidence for that. Unlikely. Or VPX and VPR genes recombined in such a way that it resulted in a VPR-like gene in SIV-ZPZ only. That's also not the case. Or the VPX gene was lost upon the genesis for a different reason. And so Mike looked at that, and this is the genome organization. Here's the VIF gene, here's the VPX gene, and here's the VPR gene. And so when you lose that, what happens is that the VIF gene gets a different carboxy terminus. In fact, there is a 60 to 75 base pair region here that's brand new, that was basically generated de novo. So why is this important? This is important because Apobec, which is one of these restriction factors, very potent, it incorporates uracil into the nascent reverse transcribed viral genome and screws up the viral genome that way. It gets into the particle and basically renders the virus non-infectious when it comes out. Well, viruses have evolved a counterattack, and the gene product in the virus that counteracts Apobec is VIF. It finds Apobec in the producer cell, binds to it, and brings it to the proteasome for degradation. So VIF is in charge of counteracting Apobec. So we were wondering whether the loss of VPX in this case had something to do with counteracting Apobec, and whether this was in fact driven by selection for changes in VIF so that the chimp VIF could actually, the red cap-mangabe VIF could adapt to work in chimps. And so Mike Emmerman looked at that, and so he looked at the ability of the red cap-mangabe VIF to counteract the red cap-mangabe Apobec, and that works great. But as you might imagine, the chimp VIF doesn't work so great. So now you go and check what's happening in chimps. And so here, obviously, the chimpanzee VIF is very active against the chimpanzee Apobec, but the red cap Mangabey version is not so active, had to adapt to counteract 3G and 3D. There are different versions of ApoBec, but 3G is the most important. So clearly, adaptation was necessary for this recombinant virus to effectively infect chimpanzees. And so the reconstruction of the C-terminus of the VIF protein, and there are also some changes on the N-terminus, were necessary to render the red-cap Mangabey VIF into an efficient counteractor of chimpanzee ApoBec 3G. So why didn't the red-cap Mangabey and mustached viruses infect humans directly? Well, maybe going through the chimp as an intermediary host was to adapt stepwise, go from these monkeys to apes, and then it's easy to go, or easier to go from apes to humans. And so Mike asked whether chimps provided sort of an intermediary stepping stone along the adaptive road to humans. And so he looked at that, and obviously human VIF, HIV-1 VIF counteracts human ApoBec. Chimpanzee VIF counteracts human ApoBec very well, but red-cap Mangabey VIF does not. And so this is a major hurdle, but when you look in chimps, the differential is much smaller. So the red-cap Mangabey VIF counteracts the chimp A3G better than the human. So the adaptive hurdle of going from monkey to chimp lowered the bar, so to speak, to go to humans. A direct cross-species transmission would have been more difficult. Now, you know that there are, I think, 8 or 9 different versions of HIV-2 out there now. These are 8 or 9 different independent transmission of Mangabey viruses to humans. So what is happening here? And we worked with Marcel Ooms and Viviana Simon on that. So they looked at the VIF protein. This is a model of the VIF protein structure, and there is a surface loop. And it has been known for some time that one residue 128 in this surface loop, is the region where the HIV-1 VIF hitches onto. And the neighboring residue is also where the African green monkey VIF hitches onto. And people mutated this, and it didn't work with HIV-2 or Mangabey. So Viviana and Marcel looked at that, and they found that it wasn't just a totally different region in VIF where the Mangabey VIF latched onto. It was the residue right next door. So this surface loop is a conserved binding site, although HIV-1 and HIV-2 utilize different residues. But it is something that the VIF proteins latch onto. Now, when you mutate 129, you lose the ability of the HIV, actually 128, you lose the ability of HIV-1 VIF to work. But it so happens that the Mangabey VIF just can interact with 129. And so it just so happened that ApoBec had this surface loop where Mangabey virus VIFs could latch onto and counteract this. And that explains why we have multiple transmissions of Mangabey viruses to humans and many, many fewer other SIV transmissions from apes or monkeys. Now, interestingly enough, this position, 129, in the gorilla ApoBec is a Q, which would eliminate VIF binding. And so we think that perhaps that's the reason why gorillas acquired their infection only once, whereas humans acquired it twice from gorillas and twice from chimpanzees. So ApoBec clearly has played a major role in these cross-species transmissions, really being a major hurdle going from the red-cap Cercopithecus viruses to SIV-ZPZ, being a hurdle going from chimps to gorillas, not so much from chimps to humans. Here we don't know yet, and clearly not, this particular restriction factor has clearly not been, a hurdle for the Mangabey viruses. But there are other hurdles, and another hurdle is tethering. So what does tethering do? Tethering sits in the membrane of cells, and it interacts with enveloped viruses, not just HIV, but others as well, and it tethers them physically to the surface of the infected cell membrane so they can't get off and infect other cells. And so, again, viruses have evolved counteractions And they can either use the VPU protein or the NEF protein or even the ANF protein to counteract tethering. And again, the notion is you find the restriction factor, you bind to it, and you try to get it degraded as fast as possible so they cannot exert the antiviral activity. So when virus went from monkeys to chimps, could have either used VPU or NEF to counteract tethering, but chimpanzee virus decided to use NEF. When it went into gorillas, same thing. The NEF protein is the accessory protein that counteracts tethering. But there was a problem. NEF interacts with the cytoplasmic tail here. In humans, for reasons that aren't clear, there's a 5 amino acid deletion in this tail, probably due to some other viral pressure, that abrogates NEF bindings. And NEF no longer can bind to human tethering. And so these viruses, as they made their ways into humans, had to think of a different solution. And for the Pseudomengabe, it's actually the envelope that counteracts tethering, at least for the more epidemic strain. But for the various ape viruses, really only one succeeded in generating a good anti-tethering antagonist, and that is the M group with its VPU. The N group also adapted VPU, but lost some other functions, so it's partially screwed up. O and P never could turn their VPU into an effective tethering antagonism. And we think that that explains in part the epidemiology we see, and we think that this adaptation of the group M VPU that also degrades CD4, but has now learned to interact with tethering here in the membrane. Two alpha helices come together, and these contact points had to adapt to do the counteraction, which is not the case in the chimp virus, where tethering is counteracted by NEF. So I've shown you that the origins of pandemic and non-pandemic HIV-1 have been traced back to different chimpanzee and gorilla communities in West Central Africa. I've also shown you that interferon-induced innate restriction factors, APOBEC, tethering, and others represent powerful barriers to cross-species transmission. However, whenever there is a hole in the host's armor, you can be sure that a virus finds it and takes advantage of it. And that passage through apes probably has lowered the adaptive hurdles for at least some of these restriction factors. So let's switch to malaria. Just a little bit of background. It's obviously a parasite. There are 5 different species of Plasmodium that can infect humans. The 2 bad ones are Falciparum and Vivax, but there's also Ovale malariae, and this is a monkey parasite, nosai, that zoonodically infects humans in Asia every year. Malaria in many ways is as bad as HIV. It infects millions of people. It causes 1 million deaths in kids in Africa. And about, I think, a quarter of the human population is at risk for it. And it is a major burden to health, society, and economies. And obviously, in contrast to HIV, malaria requires an intermediary vector, in this case, the Anopheles mosquito. Does size matter? Size of the genome? Probably. For an HIV virologist, going from 10,000 bases and 9 genes to 14 chromosomes and 5,000 plus proteins is a little bit of a jump, and we are still adjusting. But at least the malaria genome is not as big as the human, although it is bigger than bacteria. And so what does the extra genome size buy you? It buys you a very complex life cycle. And you all, in your textbooks and various courses, have learned about the malaria life cycle, which takes place, the sexual reproduction in the mosquito, and then it goes into the human host, where it first goes to the liver and then to the bloodstream, to the erythrocytes. And if you were to sort of take part in creation and you were to talk to God, and God would want different concept sheets for different solutions, and you would present this, he would say, "Are you kidding me? This will never work." But it does work, and in fact, these parasites are hundreds of millions of years old, and so however complicated this looks, they certainly have figured out a way to make this work. Now, luckily for us, since we deal in fecal samples primarily, there is a secretion of parasites and their DNA from the liver stage into the bile and into the fecal sample, and that's why I'm speaking here, is because we can detect it in fecal material. So before we started this, this would be the phylogeny of malaria parasites as it was known at the time. It's a little bit different, yeah, although similar to HIV. So like there's an HIV-1 and HIV-2, there are two different groups, very different groups of primate parasites infecting humans. There is this group here, where falciparum is in, and then there is the other group where vivax, nosai, malaria, and ovale are in. And they're very different lineages. And some time ago, someone suggested to call this lineage a separate subgenus and call it Lavarania. And we found that very helpful to us, as you will see in just a little bit. Now, one ape counterpart was known, Plasmodium reichenauii. It was isolated 40 years ago by the CDC from a chimp that was imported somewhere from the Lake Edwards regions in East Africa. And they characterized and sequenced this particular parasite, and this is its position with respect to falciparum. These other parasites are all infecting Asian macaques. So the origins of the human Plasmodium parasites, for the longest time, people thought falciparum started in Africa, has been there for a long, long time, has co-evolved with human and ape ancestors, is at least 7 million years old, and then somehow spread out of Africa into the rest of the world. Conversely, vivax, people have always thought it started in Asia. The reason for that is that Asian monkeys have, until recently, had the closest parasites, and then spread toward Africa. And the greatest genetic diversity actually is in Asia, so that made sense. Both of this is wrong. And if you forget everything from what I said today, remember this. This is wrong. So how did we get into this? Well, it turns out 100 years ago, people were already studying ape parasites. A bunch of different investigators working in the Cameroon and in Sierra Leone were interested in human malaria, but at the same time saw the apes being kept as pets or in the wild and wondered whether they would have similar parasites. And in fact, they did, although at the time, of course, they had to resort to microscopy as the only way of identifying them. So they found a counterpart to falciparum, which they called reichenauii. They found a counterpart to malaria, which they called rhodani. And vivax and ovale are different morphologically to differentiate, so they found something similar in apes and called it Schwetzi. They even did transmission studies. They gave these parasites, took blood from apes and gave it to humans, and took infected human blood and gave it to apes. Would, of course, not be possible today, but was interesting. Malaria is not HIV. You can cure it after you have it. So it's not exactly the same thing. But they found that falciparum or the ape version of it were not transmissible. However, the others were. Unfortunately, nothing was kept, nothing was frozen. There were no isolates for these two, and only a single isolate from that one chimp that was imported as far as the falciparum-like parasites is concerned. So we started with our ape fecal samples, targeting this time the mitochondrial genome of Plasmodium, not humans, went after the cyto-B gene, which is what most people do, and screened. And here, same story, same dance. I'm showing you chimps and gorillas. Yellow means positive, and you see that there is lots of Plasmodium infections throughout the 4 chimpanzee ranges, not in bonobos, the second chimp species, also present in eastern gorillas, in western gorillas, but not in eastern gorillas. And the prevalences are quite high, and again, these are fecal prevalences, so you can imagine the blood prevalences are even higher, up to 50% in chimps, here in West Central Africa, up to 40% in western gorillas. So when we then characterized these parasites on the molecular level, we had a surprise. Instead of seeing these 2 lineages, human falciparum and the reichenauii from chimps, we saw 6. And these clustered as if they represented 6 different species of Plasmodium, and that's what they most likely are. 3 of them in red were specific for chimps. 3 of them in green were specific for gorillas. And when we looked where human Falciparum fell into this whole family tree, it fell into one of the 3 gorilla species. And when you looked at the sequences of the human parasites, they all coalesced to a single common ancestor, which fell within the radiation of the parasites from gorilla. And that's the same thing as what I just showed you for HIV. That means a cross-species transmission, one time, of a gorilla parasite to humans. So chimpanzees and western gorillas each are naturally infected with what we call Lavarania species. These parasites exhibit strict host specificity. We never saw a gorilla parasite in a chimp. We never saw a chimp parasite in a gorilla. And we saw only one evidence for one cross-species transmission. These infections are highly prevalent and widely distributed and often comprise mixed species infections. And humans acquired Falciparum from a single gorilla to human transmission event, as I said. Now, what about vivax? This is the epidemiology of Falciparum. This is vivax. It's basically identical except for Africa. No vivax in Africa. Why is that? Well, it so happens people in Africa, indigenous people, have a mutation that allows them not to express the receptor for vivax on the surface of their erythrocytes. It's called the defi negative phenotype. The defi antigen is the receptor of vivax, and in these people, they have a mutation in the promoter that prevents expression of the defi antigen on the surface, and so these people are resistant to P. vivax. And so the one thing that people notice, though, is about 1% of travelers going to Africa would come back to London or New York or whatever, and they would have P. vivax. And people say, well, where did they get this infection? And so the question always was, is there a natural reservoir? Because people in these parts of Africa are clearly not P. vivax infected. So same story, same dance. We screened, and same thing again. Wild apes are infected with P. vivax-like parasites. And in fact, Ape P. vivax is less commonly detected because it's there there is a lower parasite burden, and therefore it's more difficult to detect it in fecal samples, but it's more widespread. And these are the prevalences, the fecal prevalences. We didn't find it in Nigerian chimpanzees, but that is because we only screened 126 samples. On average, we needed to screen 100 samples to find one positive. So it's a lot of fecal samples to go through. But the prevalences that we did find, especially in the central chimps, are on the order of human population in hyperendemic regions. So an 8% prevalence in humans would certainly indicate that the parasite is transmitted. So then we did the same thing. We targeted mitochondrial, apicoplast, or nuclear DNA to get at the phylogenetic relationships. But because fecal samples are so difficult to find positive, this time we had to do something else. And we started to work with sanctuaries. Sanctuaries in Africa take care of orphans. Oftentimes they are located in the forest immediately adjacent to the habitat of the wild apes. So whatever mosquitoes bite the wild apes will also bite these chimps. Now they are in an enclosure, but it's very natural. This is within the sanctuary. And of course they come and get treated, and on occasion their health exam yields blood. And so this is the Sanagayong Sanctuary in Cameroon that sent us a bunch of blood samples because some of their apes, their chimps, they have chimps, looked like they were sick with malaria, in one case cerebral malaria, that then went better when they treated with anti-malarials. So this is the phylogenetic tree. In color code here, chimpanzees, green, gorillas, black humans. Same story again. Here's the radiation of the ape parasites. Here are the human parasite. Looks just like Falciparum. A single common ancestor within the radiation of the apes. So does that mean vivax is the result of a single transmission? No, not in this case. Why not? because there is no species specificity among the ape parasites. They go from gorillas to chimps and from chimps to gorillas. And so what we think is happening here is there was an ancient stock of P. vivax in Africa that infected chimps, gorillas, and humans. Humans then developed the defi negative phenotype and drove the parasite out of the human population. So it went out of Africa into Asia and other places. And so what you're looking at here is a bottlenecked population of human parasites that escaped out of Africa. Same situation in a nuclear gene. I could show you 20 other trees. They all look the same. We did look in monkeys just to be sure we didn't miss anything. Because of the lack of species specificity, monkeys don't have P. vivax. They have hepatocystis. And we looked whether the bonobos, which were consistently negative, had the defi negative mutation. They don't. So chimps and gorillas in Central Africa represent a substantial P. vivax reservoir. Human P. vivax emerged, as I said, within a species that infects gorillas and chimps. And so extant human P. vivax represents a bottlenecked lineage that survived after getting out of Africa. And obviously, this provides a long-sought explanation for the defi negative phenotype, because people always said, where is the pressure coming from for this human mutation? It would happen at the very place where the parasite was missing. And so this provides this explanation. So obviously, now we have a whole plethora of new Plasmodium species. The question is, are they still transmitted to humans? And there is evidence, Nolzai is a zoonotic agent in Southeast Asia, where it causes about several hundred clinical cases of malaria every year. That's a macaque parasite. And so we asked the question, could something like this be happening in a place like Cameroon, where wild ape populations are highly prevalent, both for the Lavarania and the non-Lavarania parasites? So we went back and screened humans in areas where prevalences were high. These are the human villages, and here are the field sites where the apes are positive. And these are the types of settings. So you can imagine humans clearly do live within mosquito flying range. But to make a long story short, there was no evidence of ape Lavarania species being transmitted from apes to humans. And that makes sense because the early transmission studies that the folks did in Africa also failed to show that. And yet, when we did go and looked in ape fecal samples, we would find all the species that we didn't find in humans, where we found either falciparum alone or various combinations. However, vivax, ape vivax, is a different story. This is not our work. This is the work by a French group published earlier this year, where they looked at captive apes, which had vivax. Here's the cosmopolitan vivax strain. They don't have that many sequences, but clearly here are the ape parasites different from the human. Here are mosquitoes that were captured close to these apes, which were sort of in a semi-sanctuary setting. And here is a parasite that a traveler, a Frenchman who worked in a Central African Republic in the forest for about 3 weeks, returned back to Paris, came down with malaria, was diagnosed with P. vivax infection. So clearly that parasite falls within the ape group, and when we took the sequence of that parasite and stuck it into our trees, here's extant human P. vivax, here are the gorilla and chimp versions, and here is the European traveler. So clearly a P. vivax can be transmitted. It isn't normally because the folks are all defi negative. But if you bring in defi positive travelers or workers, they are at risk for these infections. So the Lavarania parasites, the falciparum-like parasites, are highly species specific, not so vivax. But then if this is the case, how in the world did the gorilla precursor of falciparum manage to overcome the species barrier? We don't know that, but we know a lot of things are required for a merocyte to invade red blood cells. All these different surface molecules have to be in place and interact with surface molecules on the erythrocytes for this docking to take place and for the invasion process to proceed. And one is, you can have various combinations of these, but one is absolutely necessary, and that's the interaction of RH5 with what is called the bathogen receptor on the surface of human erythrocytes. That is universally required. And so we looked at RH5 in our ape parasites, and we found something really strange. This is a phylogenetic tree of RH5, and this is the phylogenetic tree as we expected them from other genes. And something is different. You can see Falciparum prefalciparum is always closest to Reichenauii, but it's missing here in RH5. And instead, the Falciparum prefalciparum is closely to this other species that we've named Plasmodium adleri, infecting gorillas. Whereas adleri in the normal tree would always be close to Gaboni. Gaboni is here. So what has happened here is a horizontal gene transfer by mechanisms we don't understand of the RH5 locus from prefalciparum, which is the gorilla parasite, to humans. And it's probably not a coincidence that this is in a gene that is absolutely required for erythrocyte invasion. So what we really need are comparative genomic studies. We got to sequence the genomes and see what's happening here. But is this possible? And it is possible even when blood samples are not available. And what a student in my lab has developed is what he calls select whole genome amplification. He has primers that are specific for Plasmodium, but not the contaminating ape genomic DNA or bacterial DNA in the fecal sample. And he uses Phy29, which is an enzyme that basically makes more of what you have. With these primers, he can specifically amplify Plasmodium. And he gets up to a 30,000-fold enrichment, and that's shown here. This is just unfractionated DNA, extracted in this case from a blood sample. Here's a marker. Here's your DNA. He digests this with enzymes that are methylation sensitive because Plasmodium happens not to be digested. He digests this away. This is basically the chimp DNA. It goes away, can't see anything. Then he amplifies with Plasmodium-specific primers, and he gets this. And then this is Phy29 to use in the amplification. And then he can sequence, and this is one of the chromosome, and this is the type of coverage that we get, right, down to even the telomeres. So we get about 80 to 90% coverage using this approach, and I think we will be able, certainly from blood samples, sequence these ape parasites. This is fecal material. You see it's a little bit more spotty, so we have to still work on the method. But we think that comparative genomic studies will be possible, and that we will ultimately be able to do what we did for HIV, trying to figure out where are the barriers, where are the restriction factors, what was required for these jumps to occur. So this is my last slide, and these are the take-home points. I've shown you that 3 major human pathogens originated in apes. I've shown you that the transmission hurdle for the ape pathogens was lower, but only a small subset of what could have infected us humans did infect us humans. In figuring all this out, non-invasive studies of wild apes have been key, and there's certainly, I hope I convinced you, is value in studying the ape precursors of the human pathogens in their natural hosts. I always get the criticism, why do we need to know about these monkeys and their viruses or their parasites? This is why you need to know about them. And then my last take-home point comes from my sponsor, and that is incorporate pre-banked specimens, which your repository here has in many, many versions, into your research. All you need to do is walk over to the CIFA biospecimen repository, and they will likely have what you need. So these are the people who I've worked with, obviously the folks in my lab. I've mentioned some. My collaborators who collect fecal samples, my collaborators who help with the evolutionary analysis, my malaria collaborator, which is a new field that we got into, and the various people in the sanctuaries in Gombe, et cetera. Thank you.
Probably came with and into Europe and the rest of the world through
vivax most likely came to Europe and then went from Europe to the New World and to Asia. And the old stock in Europe has died out. And people have already suggested to try to undig graves and see what we can. I'll do fecal samples, I don't do graves. Falciparum, went from gorillas to humans somewhere in Africa. There was no defi negativity, so it could spread in the human population. When that occurred, it's very difficult to say, because you can't time these events because you don't know what the real rate of evolution of Falciparum is. So there have been time estimates ranging from 5,000 years ago to 600,000 years ago, depending what assumptions you make. But there are studies ongoing now that are trying to get experimentally at the rate of evolution in Plasmodium by going in tissue culture, for just passaging it, sequencing and determining it. And so far, it's interesting. Most likely, the more recent estimates are the more accurate. So it is possible that Falciparum was introduced into the human population anywhere of 5,000 to 10,000 years ago.
There's only one introduction.
Correct. And we don't know why it doesn't jump all the time, but it doesn't. And it doesn't not only jump to humans, it also doesn't jump between the apes. And that's one of the things why we want to sequence the genomes, to figure out what are the barriers to cross-species transmission. Now, it did jump once, and that always makes you a little worried, because it has figured out something. But just like was the SIVs going from monkeys to apes and from apes to humans. What it is, we don't know.
Could you repeat the question? Because
Oh,
they're
sure,
recording.
sure.
Other questions?
Yes.
So how is the reservoir for P vivax maintained in a Duffy-negative population?
So the question is, how's the reservoir for P vivax maintained in a Duffy-negative population? It isn't. It's maintained in the apes. They're not Duffy-negative. The reservoir is in the apes. No one ever looked at apes. No one suspected apes to have it. It is that reservoir that must have provided the pressure and maintained the pressure for Duffy to go away and to stay away.
Yes, so I'm trying to figure out how a traveler.
Well, you are Duffy positive, I would assume. So you go to the forest, you have the receptor, you get bitten by the mosquito, you get infected. You return to the United States, you get diagnosed, treated, you're good. So, are you asking what kind of a threat is this?
It seems like there should be some threshold for maintenance. Say even infected mosquitoes.
I'm sure there is, but the apes are really endemically infected. So they will be sitting there. So if you get bitten by mosquitoes that are also biting apes, you will be at risk of becoming infected. Now, if you are an African person, not Duffy negative, you don't need to worry about it. If Duffy negativity really is 100% protective, that's a different story and a different talk. But if things are changing, people are moving into Africa through commerce, through travel, that are Duffy positive. They could pick up these parasites. Now, what happens if you come from Korea, not from the United States? You go back to Korea, where vivax is actively transmitted. Is it possible that introduced into this new situation, ape P vivax could spread? Is it possible that ape P vivax could recombine with human P vivax? It is more genetically diverse. It has genes we already know that human P vivax doesn't have. Would it be more versatile to adapt to a new situation? Unknown. So that's where that zoonotic threat is, I think.
So the falciparum genome, if you can correct me if I'm wrong, it evolves at a really fast pace, right? Because you see that there's resistance to antibodies, and there's like synergistic approaches applied all the time. So who's to say that over time, now the Plasmodium might have evolved and it's become distinctly humans? Do you see any transmission back from humans to apes and gorillas?
What is the rate of evolution in Plasmodium, Falciparum, and is there evidence for moving back from humans to apes? Very good question. Falciparum does not evolve fast. And what is fast and what is slow is what your reference point is. Certainly isn't like HIV. And again, the actual rate is not known because you would have to determine it experimentally, because all these calculations make assumptions, like the assumption that there's co-evolution of parasite and host. Well, that would time the divergence, but it's not true. So what are you going to put into the assumption? Can the parasite go back from humans to apes? In the case of falciparum, it has, because we've identified, and others, chimps in captivity that have human falciparum. In the wild, we never seen that. And it could be that the mosquitoes are different. But for example, bonobos are not naturally infected, but in captivity they are. They are susceptible. And as far as vivax is concerned, we don't have any data, but I wouldn't be surprised to see human vivax go back into apes.
So just like the previous European traveler was infected with malaria, venture off in the forest, and potentially mosquitoes could, transmit it again.
Yes, but they're already chock full of whatever they have. So I think it would be hard for a human parasite to establish itself. We had field sites for Lavarini parasites that were close to 100% infected. I think they are just as endemic as human populations are.
So if you only need to eliminate one apparently non-essential protein to protect an animal from vivax, why is it still in apes? And for that matter, why hasn't falciparum been driven out of Africa by a similar mechanism?
Good question. Why have apes not evolved a defi negative phenotype to protect themselves? I have no clue. It is a very elegant solution that humans have found, because they have this promoter single nucleotide change that simply abrogates expression on erythrocytes, not everything else. The defi antigen is like a cytokine sponge. It sponges up cytokines that aren't needed. You don't need them on erythrocytes, obviously, but you probably need them otherwise. Maybe that doesn't happen or doesn't work in apes. Maybe there's some other biology. I don't know. Why haven't we developed yet mutations that would protect us from falciparum? Perhaps because it has been in humans relatively recently, and it takes time for these things to work. And the defi negativity is not 100%. There are now several reports that say defi negative people can become infected. But the details is still very much in flux and being investigated. Yes?
If the falciparum made the jump very recently, how would you explain then sickly trait and its frequency in Africa? I've always assumed it was a long-standing mutation.
I don't know how long these human traits take to be selected for. And it also depends whether there's any fitness cost associated with the traits. So apparently defi doesn't have any fitness cost associated. The other mutations, I'm not so sure. I would think if this is all going to pan out, it is still 10,000 years old, 5 to 10,000 years old. Whether human mutations could arise, I don't know. Yes?
It seemed like the Ape reservoirs for both SAB and malaria were overlapping to a certain extent. Do you think that means anything, or is it just that's where it is?
So what is the potential relevance of overlapping reservoirs for SIV and malaria? We have apes that are positive for both, but in all of these field sites, except for Gombe, which happen to have no malaria, unfortunately or fortunately, depending on which side you sit, we can't do any natural history studies. So we cannot determine whether co-infection would be worse than an infection with either one or the other. That can only be done in a setting where you can follow the apes and determine whether they get sick, whether they die faster, that kind of stuff. We did that for SIV in Gombe because Gombe is habituated and has SIV, but they don't have malaria, so we can't address that. I would not be surprised if the same thing is happening in apes, that's happening in humans, and it's well known that co-infection is worse than not.
Can you take us further back the history of the retroviruses? When and where, I guess I should say, was the last common ancestor of the mammalian retroviruses?
So timing these things is notoriously difficult because the methods to go back in time are notoriously flawed. But I can tell you two things. Endogenous lentiviruses sort of give you a time capsule because they have invaded the host genome and then have become a gene that's passed like normal genes are. And the lemur lentivirus, endogenous lentivirus, is 4 million years old. And I think the rabbit endogenous lentivirus is 12 million years old. So I would think that these viruses are super ancient, that they have gone to extinction and got back in, that they've gone into the genome, out of the genome, they've been around. And you see that in the arms race between the viruses and the host and these restriction factors which are under selective pressure to evolve faster. But what was the last common ancestor of the primate lentiviruses? I cannot give you a date for that.
Well, thank you very much.
Sure. Thank you.